And we're live, right? Yes.
Yes.
Welcome,
Yes,
everybody.
Oliver. Yes, Oliver, we do this.
The signals are always ambiguous. It's like 3, 2, 1. Do we go on 3? No, we go on 2. Wait, no, wait. Okay.
I think I'm going to get one of those. We're going to get one of those clapboards. Is it a clapboard?
Yeah.
Take
Yeah.
one.
I need a very direct signal. Welcome, everybody, to another one of our journal clubs that are related to aging and mechanisms of aging and diseases of aging. I'm Dr. Medvedik, broadcasting live or streaming live from the city of New York. With me is Stephen Hill, who's over in a suburb of London, I believe, somewhere. And we're both represented by LEAF, Life Extension Advocacy Foundation, and Lifespan.io. So this week, we're going to be doing a paper on Alzheimer's, and this is a paper that just came out very recently, and it's rather unique in that it uses new methodology to both study Alzheimer's and a potential drug. So let's open up this paper right now. I'm in my lab right now. Sharing screen. All right, so the paper that we are doing, and I'm assuming it's coming in nice and clear, is a Nature Medicine paper, gain of toxic apolipoprotein E4 or ApoE4 effects in human iPSC-derived neurons is ameliorated by a small molecule structure corrector by Wang et al. And this paper introduces a couple of novel things. One is that a lot of the Alzheimer's studies, a lot of the studies on drugs have been done in mice. There's mouse models, but there's a lot of problems with those models because some of the things that are seen in mice are not seen in humans, and they mentioned some of these things, or in human beings, they mentioned some of these things in this paper. So what they're doing in this paper is they're actually taking patient-derived cells, either from people who have the ApoE3 isoform, which does not predispose you to Alzheimer's, or the ApoE4 isoform, which predisposes you to Alzheimer's. And they are then turning them into induced pluripotent stem cells using a cocktail of factors that was discovered at the Yamanaka lab, for which he won the Nobel Prize. So this is also very recent, about 10 years ago. And they're generating GABAergic neurons, which are affected by ApoE4. And using these patient-derived cells, they could now test drugs. They can now test hypotheses on human cells that are from individuals that have this mutation. And specifically, they can now mature them into neurons and see how these neurons perform. So that's really that's other than, generating a little kind of, what do they call those? Micro brains or nano brains, little brains that they, in a dish, or looking inside of a person where you actually have, in vivo structure. This is as close as you can get, potentially. I'm sure the people are working on methods to get closer, but this is a vast improvement. And the other unique thing that they do in this paper is this is related to an earlier paper that they published. They are using a small molecule, which is, I'll talk more about this, but it's a structure corrector. So what that means is I'm gonna stop sharing this screen here and show my face for a little bit, and then I'm gonna switch over and I'm gonna show you some of the key molecules that the people in this paper are discussing. Let's see, where's the ApoE4? Gosh darn it. Let's see. I'm gonna, well, let me just go here. This is not it. But while I'm here, since you're seeing the screen, this is a low-density lipoprotein where cholesterol is essentially transported in your body. And there's a number of proteins that are embedded in this, and one of them is ApoE, which is right here. And these proteins essentially dock to receptors on cells and allow cholesterol and other lipids to enter cells. So ApoE is required for cholesterol metabolism. So that's its normal function. And if you actually look at the structure, APO structure, let's put that in, images right here, very good. Okay, so if you could see that structure here, this is a ribbon diagram, alpha helix. It's got a LDL receptor binding region and a lipid binding region, right? So one end sticks into the chylomicron that you saw there, and the other one that docks the lipids, and the other one docks the receptors. And then there's this flexible linker region here. And the ApoE3 and the E4 isoforms are very similar. They only differ by one amino acid. So here are conveniently the isoforms E2, E3, E4. And you have basically a change at position 112 from a cysteine to an arginine. And that makes all the difference. That makes this E4 isoform less stable. It actually fragments in the cells. And by using a small molecule that I don't know exactly where it binds, but it's able to essentially correct the overall tertiary structure of this protein such that it behaves more like an E3 isoform, I believe. So that's pretty cool. Okay, so that's enough about ApoE4 and E3. So I'm going to stop sharing here, show my face a little bit here. And that's one of the key players in that paper is the ApoE4 E3 isoform. I'm going to introduce two other key players because the toxicity that this ApoE4 isoform confers to cells is actually via the accumulation of a variety of other proteins. And I'm going to mention these two here. If I can find the darn thing. This is probably, yeah. I'll just talk about it anyway. So there's two proteins that they mentioned in the paper. One is APP amyloid precursor protein. This is a protein that's not clear what are all the functions it's required for neuronal development, but this protein here actually is what causes external amyloid beta plaques, and it basically fragments, and these plaques accumulate outside the cell and are thought to be toxic. There are also internal proteins that build up, and this is the protein known as tau that gets phosphorylated. And when it gets phosphorylated, it forms these neurofibrillary tangles, right? And they accumulate inside the cell. And the presence of these, the buildup of these, is in this paper linked to the ApoE or epsilon 4 isoform, right? So that's the groundwork for that. So let's go to the paper. Any questions? My background here. Oh, and what tau does, it interacts with microtubules. So it's a microtubule-associated protein. That's what it normally does.
No questions yet, but I am monitoring. Obviously, anybody has got any questions or anything they want to, say, they can use the chat in Facebook, and hopefully Oliver will have all the answers.
If not, Google will probably have the answers.
Google.
Yeah. Okay. So, onto the paper. Get rid of this thing here. Oops, there it is. All right. So, what do they do here? Well, as I mentioned, they take cells from patients and they generate basically neurons, GABAergic neurons. GABA is I believe, gamma-aminobutyric acid. That's a neurotransmitter. It degenerates. I believe these neurons die off in Alzheimer's patients and a number of other patients. I believe GABA is an inhibitory neurotransmitter. But anyway, they're neurons. They signal. They're in a brain and they're sensitive to certain types of damage, as mentioned in this paper. So this first figure here has a lot of parts of these figures. It's like figure 1, A through U. It's almost getting to Z. But anyway, this figure here is essentially they're showing you how they generated these neurons, these E4, ApoE4 neurons, and basically characterize them, right? So there's ApoE3 neurons, ApoE4 neurons. So they take these cells that don't know exactly, they might be fibroblasts or skin cells. I have to take a look in the paper. I'm sure it's mentioned where they have these cells. They're biopsies, don't have patients. And they use the cocktail of factors and basically created neurons. So they want to now characterize them and show that they are indeed functioning the way they're supposed to be functioning. So this is all characterization. So these are a lot of blots. So you're saying essentially here Western blots, and these blots are just probing for proteins. Actin is a control. ApoE, E3 variant, E4 variant. And you can see that it is being expressed. There's secreted form. There's internal form. And they're showing ApoE actin. So this is full length E3 or E4. So these are full length. And what they're showing is that you have greater full length E3 versus full length E4. So this is a blot that's normalized to the actin control. And that's suggesting that it's being fragmented. And when you look at figure D, you can see down here, you can see a smaller form or fragmented forms of E4. So that's what it normally does. So, these bar graphs are just basically quantifications of these Western blots. So you have significantly more, several more fragmented E4 versus E3, which is more full length, which is more typical of the normal variant. What they look at next, EFG, I got to go alphabetical order here. More characterization. All of this stuff, AT8, AT180, PHF1, AT270 are all different, I believe, phosphorylated forms of tau. So you're expecting to see more hyperphosphorylated tau. This is control, tau5, TJ1, which is a neuron-specific protein. And you can see these bands get darker. So the antibodies are specific to hyperphosphorylated. At particular, we could look these up and see which amino acids are specifically phosphorylated. And here are just a bar graph that quantifies all of this and p-values. So very low p-values. So we're confident that this is real. So more phosphorylation seen throughout E4. So these are the neurons that they generated from patient cells. So they want to characterize these to show that they're performing as expected. Next, they do some fluorescence microscopy, and again, this is just again looking at phosphorylated versions of tau. PHF1 MAP2 is a neuronal specific marker for a microtubule associated protein. AT8 PHF1 you can see in the E4. You can see more yellow and red superimposed, so you see more phosphorylated versions of tau. Again, bar graphs showing that when they look at a field of view, AT8 cells, over MAP2 cells, over total cells, you've got more phosphorylated cells, right, within the field of view. And then they look at amyloid beta. So there's two versions, AB40, AB42. These are toxic. These are secreted in the media, and again, E4 has more. Right, so more amyloid beta protein is being secreted. This is SAPP, secreted amyloid precursor protein. More of it's being secreted in the E4 variant. What else are they looking at here? APP, mRNA. Oh, so what they're looking at here is, although more of it's being secreted, more of it is not being actually expressed or generated. And I think, let me see if I go back to this paper. So in the paper, they mention, of note, the levels of full-length APP protein. Let me go back, stop sharing here. And I read from the paper. Of note, the levels of full-length APP protein and of the APP mRNA were similar in APE4 and APE3 neurons as determined by Western blot, respectively, suggesting that ApoE4 did not alter APP expression. Okay, big deal. But they mentioned that this indicates species differences because when they look at mice, it's different. ApoE4 stimulates A beta production in human neurons, but not in mouse neurons. So this A beta, so if you have ApoE4, you have more A beta production in human neurons. So the A beta form, not necessarily expression, they go up in humans, but not mouse. So that's something that's a clear difference that we're seeing, that they notice here, right? So that kind of ties into what they've been saying, is that, using these types of systems, these iPSC systems, you can potentially generate data that's more significant when it comes to human therapeutics. Of course, we have to look at these neurons in the context of the human brain. There's a blood-brain barrier, et cetera, et cetera. So, again, this is all being done in the dish. So just letting people know here, if any drugs they introduce, they're basically putting them into a dish. So any positive effects, you have to kind of temper that with the fact that the compound is still going to have to, once it gets into a human body, it usually gets processed or not by cells in the liver, might get changed to a different version. It might have problems going through the blood-brain barrier. It might have toxicity to other cells. So, there's still a long way to go before, this thing is shown to be efficacious in humans. But anyway, so that's figure 1. Any questions about that? So they're just characterizing these neurons.
Oh, we got the questions actually. Just loading them up. Right, that's probably one for a bit later towards the end. We got asked one by Forefire from our Discord community, a question earlier in the week. Yes, we're saving them up for you, Oliver, now. But we've just had a question in. We just had a question in from Facebook. Philip Powell has asked. He says, "Forgive me if this has been mentioned. Is this a special kind of protein? What is its source? Animal or plant? Would it matter?"
Oh, what are we talking? Which ApoE4 protein?
Yeah, that I think that's what he's talking about.
Oh, yeah. It's well, it's a protein found in all organisms, mainly animals, I believe. It's used for transporting lipids, primarily cholesterol, and there's a number of other proteins that are bundled up with it, and it basically helps the lipids that are floating around in your bloodstream to get where they're supposed to go. Otherwise, and I think this is a question you had earlier, Steve, because they had cells where they knocked out ApoE completely, so there's no ApoE gene completely, and there's actually people that have ApoE knocked out, deletion, and what happens is that they have, I believe, hypercholesterolemia. They have cholesterol deposits happening all over their body, so they might be protected against Alzheimer's, but you got heart disease problems there. So, so it is important for getting lipids, getting fats, fatty acids out of your blood and to where they need to go. So that's one of the functions of ApoE. And there's different forms. When I say isoforms, basically you can have different, they're called different alleles. You have different, mutant forms, and they more or less perform the same function. But some of them, can perform the function better than other forms, and some of them, are more predisposed to being problematic, as this ApoE4 form shows. That doesn't mean that it can't confer some selective advantage in some other direction, right? Because it's always a trade-off, right? We're in a very complex environment, so the ApoE4 isoform might have some benefit early on in life, and that's why it's maintained in the population at such a low frequency, but then it becomes problematic. I believe you have a 65% higher chance than having an E3 isoform of developing Alzheimer's beyond a certain age if you have the E4 isoform. So that's ApoE, right? So I'm just going to refer to ApoE generically, but just realize there's various slightly different versions of it floating around. Okay, share screen. Back to paper. There we go. All right. So now they look at phosphorylated tau levels, and this is something. So they want to. So they're looking again in ApoE4 neurons, and they want to correlate. So I mentioned that there's these two toxic proteins. There's phosphorylated tau that builds up and forms these. Basically what you get is you get piles of proteins that stick to one another, and they basically just clump up and they can't get gotten rid of, and they're toxic to the cells. And one form is, from APP, you have amyloid beta, which is on the outside, and then you have hyperphosphorylated tau, which is on the inside. And what they want to show here, or at least what they notice in the neurons that they generated using the human-induced pluripotent stem cells, is that when they use a variety of inhibitory factors, so these are gamma secretase inhibitors and beta secretase inhibitors. Those secretases are required for the amyloid beta to basically get derived from APP, right? So there's a cleavage happens, the protein and cleaves it, the APP, and then it essentially accumulates on the outside. So they basically they're showing here that there's a disconnect between tau and APP. So it might be two separate pathways that are somehow being switched on by E4. So if you inhibit if you add these drugs, OM992 and ApoE4, which I mentioned are secretase inhibitors, there's less amyloid beta 40 and amyloid beta 42, which are the two versions, get secreted, they go down. I'm assuming this gets as far down as E3, but I don't see a control for that. I probably mentioned that somewhere. But anyway, even though they go down significantly, the phosphorylated versions of tau still are high, right? So even though you have high levels of tau phosphorylated, which can cause neurofibrillary tangles, even though you don't have the A beta being secreted. So you can have one independent of the other. It's basically what they're showing here. So that was something that popped up in their paper. So that suggests that ApoE4 might be downstream of both, and that targeting ApoE4, as they're suggesting later in the paper, using this corrector molecule, it's probably some kind of small molecule. We could look that up later. Could ameliorate both separate events downstream. All right, so stop sharing here. All right. Any questions? I just want to show my face for a little while before I share screen again. All right, we'll dive right back into the paper. All right. Now comes the interesting part, which is what exactly do all these things do to the neurons, right? They build up there, and as I've been saying, they're supposed to be toxic. So this figure 3 legend basically goes right to tells you the punchline. ApoE4 causes GABAergic neuron degeneration and/or loss in HIPSC-derived neuronal cultures. So what we have here are a field of view staining fluorescence microscopy, GABAergic, MAP2, all neurons, GABAergic neurons, E4, and they basically, what they do is in the culture they're adding these factors to get these human somatic cells to differentiate, turn into human induced pluripotent stem cells, then convert into these neurons. And as they do so, you get less GABAergic cells coming out the other end if you have the E4 version, right? So suggesting that there's that's probably detrimental to the formation of GABAergic neurons. And over here, what they do is, so GAD65 and GAD67, I think, are markers specifically for GABAergic neurons. So, this is just a Western blot, probably of the entire thing, showing that there's less of it. So, another way of saying that there's less GABAergic neurons that you have in your culture. And this is a bar graph that just basically, you could have probably hidden all of this, but there's a lot of supplemental data to this paper as well. So if you go through it, one thing that they talked about that I don't think they have in these figures is, or maybe they do in a later figure and I'm just forgetting, but they actually also, I think, recapitulate all of this work using normal cells, E3 cells that they've converted to E4 cells using CRISPR, and they've taken E4 cells and CRISPR'd them back to E3 cells. And that's pretty important because it suggests that, and they got the same results, suggesting that it's specifically the mutation in this ApoE, right, E4. There's no other hidden mutation, right, from these cells that they derived from patients. So that gives strong, very strong evidence that we're dealing with the mutation that is specific to ApoE4, and there's probably not another needle buried in a haystack that could, and that could initiate these things that are happening downstream. Although certainly there's other mutations that can predispose someone to Alzheimer's. I believe there's mutations in APP that make APP more, prone to, cleavage. But those things may be downstream. Okay. So again, GABAergic neurons, AT8. There's more the ones that are out there, I believe, are more phosphorylated, so they have more of these tau proteins that are phosphorylated. And again, weeks 1 to 5, they basically look at these cells, and this, even though these legends here on the y-axis are the same, they're actually using 2 different antibodies to look at 2 different phosphovariants of tau, and those go up over time, right? So basically, less cells emerge that are GABAergic, and also phospho-tau goes up. TUJ1 is basically a neuronal specific marker, I believe. So you're looking at p-tau relative to that phospho-tau. And right here, we're basically just looking at the data that we just mentioned, right? So you have more p-tau, phosphorylated tau. You have in E4 versus E3 when you overlay over these graphs. Okay. I don't think they did, so let me just stop sharing. Again, there's a lot of supplemental data here, and I have to dive into it. So they talk about neurodegeneration, and what they're showing here is neurodegeneration. You have less neurons being developing into GABAergic neurons. I don't know if they looked specifically at apoptosis or how this neurodegeneration exactly happens. They might mention in the supplemental, I haven't read that. There's a lot of stuff there. But I don't recall seeing anything regarding apoptosis if that's being switched on later. Anyway, share screen again. Go back. Okay. Figure 4. Alzheimer's disease-related pathologies in human ApoE4 neurons, and they say E44 because these are homozygous, meaning both copies are knocked out, or mutant actually, not knocked out, are specifically induced by ApoE4. So they look at some of these pathologies. So what are they looking at here? So they're looking at fragments of ApoE, right? So they're looking at, I was thinking, didn't they do that before already? Is there a difference here in these? Let's see. Quantification of full-length ApoE4 or ApoE fragments. The lysates of neurons derived from ApoE4 HPICs or from isogenic ApoE3. Okay. They look at in figure 1. Let's go back. Sorry. Is that a difference? Neurons generate more human ApoE4 neurons. See. This one, there's a difference between those two. But anyway, so you get more amyloid beta being secreted, amyloid beta 42, amyloid beta 40, more fragments of ApoE. The E4 variants, the full-length ApoE is less of it, obviously because it's getting fragmented. They're looking at tau phosphorylation. So those are basically all of the things they're looking for. And all of that goes up, right? And these are in GABAergic neurons. I don't know specifically why GABAergic neurons are more prone to this. And, you certainly got a lot of other neurons that use different neurotransmitters. I don't know, maybe there's a neuroscientist out there who can tell me why specifically GABAergic neurons. ApoE4 confers a gain of toxic effects in HIPSC neurons. So one thing that they wanted to kind of clarify in this paper is it's not necessarily a loss of function. So, you know, there's two types of mutations you can have. One is a gain of function, meaning the protein itself is doing something toxic, versus the protein is just not functioning, right? And something happens. So they want to show that, you know, that this is actually a gain of function, right? Gain of toxicity here. So what they're looking at here is basically everything we looked at before. You know, we're looking at phosphorylation, we're looking at the number of GABAergic cells that are being formed, and let's see, what are they using here? So they're using cells that are completely knocked out for ApoE, right? So these are ApoE homozygous double negative cells, right? And what do they do here? So I believe that they introduce either E3 or E4 using a lentivirus. Is that right? They use a virus. Yeah, lentiviral vector. And see what happens, right? If they put put this back into these cells, because not having ApoE, right? You have a complete loss of function, right? So if the thinking here, the logic is that if you put back ApoE4, it shouldn't make a difference if it's a loss of function, right? But if it's a gain of function, it should do something. So that's what they observe, right? So p tau total, IE3, E double negative, same. Phospho tau, same. E3, same. Amyloid beta, everything's basically, double negative E knocked out ApoE looks the same, like, for what they're looking at here. The phosphorylation of tau, the release of amyloid beta plaques looks like the E3 isoform, right? So you basically have cells that, for the purposes of these characterizations, are normal, right? And then they add, lentivirus vectors which express either the E3 or the E4 isoform. So this is basically a Western, I believe, some fluorescent antibody. This is your control, and here is your one of your phosphorylated versions of tau. And so the ApoE relative to E3 levels, when they express it, are about the same, but the phosphorylated phospho-tau goes up. I think it goes up as much as what they showed before, but it does go significantly up, right? So at least for the phosphorylation status, that is served. And then they look at, in the field of view, GABAergic neurons. So here, what they're doing is they take, so I believe that these are already neurons, and I'm just trying to see when they transfected these cells with these lentiviral vectors. Okay, so these are already neurons, so they're not developing. So these are neurons, and they transfect them with, either control, which is an empty lentivirus with no plasmid. This one with 3, this one at E4. This is GABA and DAPI. DAPI stands for nuclei, and then GABA stands for obviously GABAergic neurons. So you can see in this field of view, you got a lot of nuclei, but really very little GABA staining. So what they're showing here is that, these cells, there's less of them if you basically, in the field of view, if you transfect them with a lentivirus expressing E4 versus E3 or the control. And when they look in the media, they notice more amyloid beta, significantly more that's accumulated in the media versus E3 and the control, which is nothing. There's a little bit, but, well, it's a slight increase. But again, these bars indicate they did the statistics. It's significant. So p-values aren't as high as low as they were in the other ones, but still p.05, which is kind of like your baseline of acceptance, right? It's 95% certainty, p.01, 98% or something like that. So anyway, acceptable statistically. This is figure 5. And moving on to the last figure, this is where they actually work with this small molecule structure corrector that they call pH002. I don't know what pH002 is. Some sort of molecule that's probably not very charged. They can go through a plasma membrane. And as we see here, they basically do all of the same characterizations. They're looking at the amount of GABA cells in the view. They're looking at the secretion of amyloid beta. They're looking at phospho-tau, all as basically markers for cells that are going to, degenerate. Right? So here we have cells expressing E4. So these are just a Western blot showing fragments. Right? These bands here are your smaller, chopped up version of E4, which is believed to be the toxic form. If I'm not mistaken, might be, maybe, I'm not sure. And what they're doing here is sulfoxide. So, and this is with nothing. So you can see right off the bat that, and compared to E3 control, that you have less of it. Once you add this pH002, whatever amount they added here, I'm not sure how much they added here. Hard to say. Well, it's not hard to say. They didn't mention it somewhere there. But anyway, they add in a certain concentration, and I think below they actually do a gradient of different concentrations of pH0 to 10 nanomolar to 100 nanomolar. But you see it's having an effect that's ameliorating all of these characteristics that are connected to GABAergic neuron degeneration. So this is just bar graphs of the lot of the gels that you see here. Again, they're looking at phosphorylated versions of tau. So E4, AT8, GAD67 is GABAergic neuron marker. So you have less of them, obviously, but whatever's there, you have more phospho versions of tau. E3 DMSO, obviously you have less. E4 DMSO, lots. E4, still a lot more than E3, but there's an attenuation. So it is attenuating phosphorylation. It is attenuating also the secretion of amyloid beta. So you add E4 and you have a decrease. What is this here? E4. I think this should be E33. This is a typo because this is E33 here, right? So that would make sense. So the levels go down. Actually, the amyloid beta levels are more impressive than the phosphorylation levels. They go down as much as E3 versus E4 and DMSO. And here they just do a variety of concentrations, showing, this is amyloid beta in picograms per mil, and this is the ratio of p-tau to total tau. And again, it goes downwards, and you have more GABAergic neurons in the field of view going up, using this GAD67 marker when you add this pH002. So, stop sharing. That's the paper. I think, to me, it's pretty cool having this, having this technology available where you can now use human-induced pluripotent, generate human-induced pluripotent stem cells to test drug compounds. They're testing these just on naked cells. I'm just calling them naked because it's just purified. They mentioned in the paper that that these are very fairly clean cultures. Like over 90% of the cells were these GABAergic neurons. But people are also talking about these human on chip type of assays where you would have different cells, right? Because when you inject a drug into a person, it doesn't just target the neurons; it goes to the liver. It has to pass the brain barrier and all this other stuff. And there's other cells there, and and I mentioned kind of three things that a drug would have to be capable of besides just ameliorating, what you're seeing in a dish. It's gotta do what it has to do. So this is a baseline. It's got to do what they show in this paper, but it's also has to go into the right location, which is the brain. It can't be toxic to anything else, so it can't induce a heart attack. It can't bind to ion channels or something like that, which that has to be screened for. And, it has to be relatively stable in the body, right? It can't be metabolized to something else. Or if it is, then hopefully it's an active version, right? So having a more complex system on a human on a chip would really speed up the development of drugs. But this paper here, I think, lays some of this groundwork, right? Because now we're switching over to human cells in a dish, and we're already noticing some differences, some key differences between mouse cells or mouse models and human models, namely what I mentioned earlier, which is they say, you have this drastic reduction of amyloid beta levels, and what is it here? Hold on a second. There are no significant ApoE4 stimulates amyloid beta production in human neurons, but not in mouse neurons, right? So that's the difference there. So yeah. So besides this small molecule, which is really cool, if it could basically have somebody mimic the E3 isoform, then that's great. As long as it doesn't do anything else, right? Because again, they didn't measure if you do add this molecule, how does it affect the ApoE3 version from doing its normal job, which is delivering lipids to cells. Maybe they mentioned that in an earlier paper, right? Are there any side effects? So there's a lot of things that this molecule has to, prove itself before it's, taken by people. But I like this paper. I think they do a lot of assays here that kind of lay the groundwork for future drug discovery. That's my take on this. Whether or not this drug molecule will actually, help, whether it's more effective as a preventative, could reverse effects of Alzheimer's, I don't know. But that obviously begs the question. So I heard that there were some other questions.
There is, yeah. We have one from Forefire, who's from our Discord community, and they've asked, is there any observed effect, hypothetically due to existing defective protein breakdown mechanisms being able to catch up with the buildup of the broken protein once its production is stopped? Or is the benefit merely a cessation of continual unplanned cell death?
Is this in reference to the small molecule and what it does?
The overall effect, yeah.
Well, I would. Well, I mean, what it implies here, obviously, is a ceasing of the accumulation of phosphorylated tau and release of beta amyloid, and so it halts it. Whether or not there's a reversal and that allows the body to catch up, I mean, this assay can't really tell you that. Like I mentioned, these are just cells in a dish. There's no macrophages there. There's no glial cells, which perform a lot of functions in the brain, like clearing out these plaques. So I don't know. I mean, this is something that you can probably I mean, right now what's really cool is they have these these mice that are immunodeficient, and you could generate humanized mice, right? So there's mice with human neurons, there's mice with human blood system, right? So you can generate mice with human neurons that have these defects. You could potentially see if that happens, right? Do you have a decrease in plaques after you introduce this molecule? Does it allow bodies, other systems, glial cells that act as macrophages to actually come in and clear this stuff up, and can there be a reversal? I'm thinking maybe, just because there's been other papers, I don't think we covered them, but there's been other papers where if you stimulate macrophage activity, these are in mouse models. For these mice are prone to basically having APP that's mutant that generates a lot of amyloid beta. So these are basically transgenic mice, and that if you add compounds like interleukin-33, which stimulates macrophage activity, they have a reversal of effects, suggesting that you can you can clear this stuff back out again, assuming it hasn't gone too far. So I'm being optimistic, and I'll say, potentially, yeah, you might have. It's a lot of maybes, right? I can't say for certain.
Well, yeah, I mean, and macrophage polarization is also a factor as well. There's been a number of studies showing that changing the polarization of macrophages from M1 to M2 type tends to address quite a lot of issues, particularly with plaques in the arteries, for example. And Alzheimer's has been likened to kind of heart disease in the brain, in a way. There was a paper last year that made a very direct comparison of the two diseases, and essentially saying that Alzheimer's was very much like heart disease. So macrophages are likely to play a role in that. That's the question. So what we've got to do is obviously test it in a proper physiological environment, and mice just aren't going to cut the mustard, let's face it. I'm getting increasingly disillusioned with mice, unfortunately.
Unless it's something funky like humanized mice.
But again, it's the old caveat, though, isn't it? People are still going to say, yeah, but it's not actually the environment you find. Just to go on a slight tangent, do you think that our ability to model in silico and such like organs on chips like Athena, do you think they'll potentially reach the level where they can be meaningfully used?
I mean, in theory, there's no reason why they can't, right? I mean, that's what people are aiming for, and that's what people are investing in, basically. I don't want to use the term gambling on it, but there's theoretically no reason, right, why that can't work, why you can't basically simulate all the most important functions that you need for drug discovery on a chip for certain disease states. So yeah, why not? I mean, that would be, if you can optimize this, then you can automate the heck out of this. You don't need mice, for these types of, at least these drug screens, these drug trials, and it would really, really speed up things. So this paper is, in a sense, a step in that direction, although it's just single cells that have been induced in dish.
Yeah, I mean, the systems pharmacology is getting more and more sophisticated in how it models things, and of course, deep learning is another technique that seems to be starting to be used more and more. But yeah, I definitely think whilst mice, they do have their pros. I'm not saying that mice are not useful, but I think as we sort of start trying to cut to the chase with human aging diseases, I think we're going to definitely need to find better analogues than mice. Some people have suggested things like dogs, but even dogs they're not really that similar to us, or even monkeys. And of course, they're highly expensive to run any studies with primates and things like that as well. So we basically need to do this. I think really, it's going to be a challenge, though.
Yeah. Well, everything's a challenge, I guess. Everything that's important is a challenge.
True. We've got another question here. Where is it? I've lost it now. Jared has asked. He's one of the people from Discord. He said he'd like to know a little bit more about the delivery methods to get past things like the brain-blood barrier. So if these small molecules can't get past the brain, the BBB, I'm just going to call it BBB.
Yeah.
What are our options? I suppose intracranial injections and things like that are not out of the question, are they?
Yeah. Well, obviously not. But if you want to, you obviously want to kind of avoid that, or potentially, One I do know that there's some papers out there where they looked at focused application of ultrasound that would temporarily kind of, I don't exactly know what the mechanism is, but it somehow jiggles the cells to, make the blood-brain barrier more permeable. So when you deliver the drug and you use this application of ultrasound, which is otherwise fairly harmless, it's about the same frequencies I think people use for regular ultrasound. They use it on babies, but if they focus it on a particular area, that makes it more permeable. I don't know how much more permeable, but in that case, if you deliver the drug and it's in the bloodstream, it can have it pass through. I know that's one field that's being studied, to get past it. I'm not familiar with all the other ways. There might be some other nanoparticle approaches that do something, package molecules, something pass through. But the ultrasound one is the one that I've kind of read a paper or two about.
Although, as I say, the very worst case, we could get a big old needle and jab you in the brain. It's a bit crude, I've got to say. You can go up the nose. That's a favourite. But at the end of the day, if it's grim, but what are the alternatives? If I said to you, okay, well, we can probably stop you getting Alzheimer's, but we're going to have to stick this big old needle up your nose, or would you prefer to get Alzheimer's? I think it's a no-brainer, isn't it? Really, you're going to take the needle.
Yeah. Well, potentially there's going to be easier ways to do it, but of course, needles obviously going right directly, in is the most direct approach. But I have faith that we will figure out ways to, if not ultrasound, which is showing some promise, some other ways to
well, here's hoping. Here's hoping, Oliver. A bit of a confession of mine is, you probably don't know this, is I've got a bit of a phobia of needles.
Oh.
In 1982, I actually got very, very ill from pneumonia, and pretty much almost died when I was 7. And I had to have a very long course of, penicillin. And a kind of, no one likes needles really, but it kind of didn't really endear me to them. So hopefully they'll crack that, because I don't really fancy a giant needle.
Yeah, well, as a kid, I used to be completely freaked out as well. I remember once in the doctor's office, once the needle came out, I actually dived under the examination table for a while to get me. But I've gotten over that now. I donate blood on a regular basis, so
Yeah, I've manned up, as my mum would tell me to do. Man up and get yourself. This is my mum. She said, man up and go and have a blood sample taken. For goodness sakes, it's only a little needle. But it's easy for someone else to say that when they're not the one that's having the needle stuck in them. But I just don't look now. That's my policy. Just don't look. It's somehow better, isn't it?
Well, let's face it. Nobody really wants to be injured unless they have a big streak of masochism.
Well, yeah, true. Although there is a new device that's being developed now that can draw blood without actually a needle. I don't know if you've seen that, but you put it against the skin and it somehow pulls the blood through the microscopic pores. But you can, I'll send you the link to that later. But it's quite fascinating.
Without shearing cells or anything like that?
I don't know. Obviously the skin has got lots of micropores on it, and it must somehow open those pores up, stimulate them in some way, and then use it. I'm trying to think what company it is that's developing it. It's a big company. It's not. It might be Google. It's someone like Google, or it's someone big, or like Apple, or like the medical side of those companies.
Okay.
It's not
something like that sounds really, interesting to say the least.
Yeah, we should do a journal club about the sort of technological side of things, perhaps like medical wearables and all the interesting things that are being developed as well. Because I find them as fascinating as the actual research itself sometimes. Hmm. So, yeah. So the conclusion here, though, with this paper is pretty exciting. Obviously, I've got to be appropriately cautious because it's not a done deal. But what I found really striking in the paper was the fact that people with the APO4 gene variant were, if they had one copy of that gene, they were like twice as likely to get Alzheimer's, which is significant. But what was really scary was if they got two copies, they were a staggering like 12 times more likely to get Alzheimer's. And I think that's kind of crazy. But as you said earlier, APO4 probably conveys some kind of an advantage when you're younger.
Yeah.
Good old nature, eh? Because nature
Well,
wants to get you to reproductive age, and then it doesn't care.
well, that's something we could have another journal club on various evolutionary theories of aging. One of them, obviously, being antagonistic pleiotropy, right? Something that
Yeah.
gives you an advantage early, where selective pressure is the highest, is going to obviously, more cortisol makes you more agile and stronger and more impervious to pain, but it also degenerates your tissues faster, right? So it's a trade-off.
Yeah, it's interesting. And we should probably invite someone like Josh Mitteldorf as well to join us for that one. He's got some pretty interesting ideas about evolutionary biology.
Yeah, I would love. Well, I'll talk to him about it. He was very cordial when I met him at one of the meetings. Although I have to say, I read the book and I disagree 100%.
Oh, that's what's great, though, isn't it?
Yeah.
You can agree to disagree, and still have a great conversation about it. Because I mean, we're all on the same team here. We're just coming at things differently.
Right. I mean, there's really two fun. Well, I guess there's two fundamental camps, but I'm definitely with the camp where aging is a function of entropy, non-programmed, and then you've got the other camp, which is no, it's programmed in. But I don't see the evidence for that.
I kind of sort of fall in the middle somewhere because I'm contrary like that. I make no bones that my favourite pet theory is hallmarks of aging, and for me that's why I like hallmarks of aging, because it kind of resolves the two extremes and says, well, it's not damage or program, it's kind of a bit of a mix. So you've got like the epigenetic sort of element, which is arguably programmed to a certain extent.
I hear
but
what
then
you're
you've very much got the damage.
Right, I know what you're saying there, and I think that even though that's the case, I would still put that in the non-programmed camp, because people who argue with the programmed theory of aging will say aging evolved as a benefit to populations. It's not, they go further than that. I don't see the data supporting that.
Yeah, I mean, we could have a good old chat about that, and we could also obviously touch the big subject in that, and we could also talk about, are there actual genes that are designed to kill us? What are they called? Deb Pro Death genes. That are specifically designed to kick in at a certain point to destroy us. Some people think that there are actually destruction genes that are built into us to kill us off. Are there?
I think entropy does a fine job. I think all you need to do is wear down the longevity promoting genes, and once that goes, death is inevitable.
Yeah, it's mostly a maintenance issue, and once you lose that ability to maintain homeostasis, you are you're up a certain creek without a paddle, basically. I'm sure you know the creek.
Yeah, you equilibrate with room temperature rather rapidly, and once that happens,
but there we go. So yeah, that's another one to see if see for a future episode. Let's get stuck into a bit of evolutionary biology, and we'll see if it doesn't end up with us falling out with each other. I'm sure we'll be lovely. Josh is quite polite anyway.
I'm sure it'll be cordial.
I'll be like, whoa, break it up, guys, break it up. Come on now, it's not worth it. It's just a theory. I don't know. So yeah, in conclusion, I think that was a pretty good paper anyway, pretty promising. As you say, it does lay the foundation for better models in Alzheimer's, and that's what we desperately need. And yeah, translation. I mean, you know, how do you think it would be? I mean, we can technically do it now, can't we? With CRISPR, we could technically go in. How easy would that be? I mean, you know, obviously very difficult, but
the question is obviously how many neurons would you need to target, right? Like, is there, you know, is there a certain minimal level of neurons, you know, and then, you know, what vector are you going to use? You're going to have to use a vector, probably some kind of modified herpes virus vector or something. I don't know. There's a lot of technical challenges. In theory, of course, in theory, it's possible, right? We have the tools, but applying those tools in an efficient manner, which will enable a cure, is the problem, right?
So I suppose, you know, at the moment really we've got not much choice really, but to press ahead and do everything we can safety wise, and then you know, just give it a go and see if we can do it. Sometimes there comes a point where you've just got to try these things. Otherwise, we're not going to get anywhere. And you know, let's hope we can hope we can end Alzheimer's. I'm certainly optimistic that we probably can and almost certainly will do. It's just a question of when, isn't it? Really.
Well, look, if we weren't optimistic about it, that this is going to happen, we wouldn't be having a journal club and we wouldn't be doing any of these experiments, right? So obviously everybody that's doing these experiments is optimistic that it's going to work. Otherwise, you might as well just, I don't know.
It'd be a bit strange, wouldn't it, if we were as advocates, if we didn't really believe what we were doing was actually going to work or might work. So there we go. Good point. Touche.
Is there such an advocate? I don't know. An advocate. It might be in a Franz Kafka novel. I don't know. An advocate who doesn't really believe what he's advocating. The jaded advocate, although he still gives his all. I don't know.
I don't think that's possible. that would be a paradox. And those sorts of things tend to make the universe implode. So no, probably not.
Okay, I'll leave that to the psychologists. We'll stick to aging. All right.
Yeah, we'll deal with that. That's enough for now, isn't it?
Okay. Well, take care, everybody.
Right.
See you next time.
So thanks for everybody who's joined us today, and just to remind everyone that we appreciate the support of our Lifespan Heroes. If you'd like to check that out, it's www.lifespan.io/heroes. And if you'd like to see us do more of these and you enjoy it, then consider supporting us by being a Lifespan patron. And that's it. We will let you know what the next episode is next time in due course. And thank you very much for everyone who has joined us today. Thank you. And a salute from Oliver. There you go. Thank you very much.